Welcome to this Sigma-Aldrich seminar series. Today's topic is on reference gene validation. My name is Anders Bergqvist. To begin this presentation, we understand that unknown processes of the biology of our samples and technicalities of sample handling contribute to confounding variations in our experimental measurements. In order to minimize confounding variations, we aim to normalize data in various ways. An often used approach is to normalize data against an endogenous reference gene. Throughout this presentation, we will see that the purpose of normalization may benefit from some consideration, and it may not be as obvious as one might think. A reference gene needs to be validated on the specific assay for which it is going to be used. There is no such thing as a universally valid reference gene. Furthermore, using several reference genes have many advantages compared to using only one reference gene. However, there are also factors limiting the optimal number of reference genes for an assay. In passing, we would also like to mention that there are alternatives to using endogenous reference genes for normalization, including using estimates of total RNA. Depending on the specifics of your particular assay, this or other alternatives may be competitive options to improve assay precision. For this presentation, we will use two different reference gene validation algorithms, NormFinder and geNorm. Both of these are available in the GeneX data analysis software from MultiD. Often the intended purpose of normalization is to make data comparable to a one-on-one relation to sample cell count. In this example, we have illustrated two genes of interest in yellow and orange, and two reference candidates in turquoise and blue. Suppose that two samples contain equal numbers of cells. One sample is where the cells have undergone some treatment, and another sample where the cells have received no treatment. As we measure the gene expression in the two samples, a gene of interest that shows equal expression level in the two samples we consider not responding to the treatment. A gene of interest that shows different expression levels in the two samples we consider regulated due to the treatment. Likewise, a reference gene candidate that shows equal expression levels in the two samples we consider a stable reference gene, good for normalization, and a reference gene candidate that show variation in expression levels in the two samples we consider an unstable reference gene that is not appropriate for normalization. The challenge, of course, with this setup is that we do not know a priori how many cells are present in each sample. Consider this extreme example. Due to differences in the biology of the treatment response or differences in the technical handling due to the treatment response, suppose that the treated sample contains twice the number of cells as the untreated sample. With this factor unknown to us, we may reach an opposite conclusion as to which gene of interest that is regulated due to the treatment and which reference gene candidate that is stable under assay conditions. An analysis approach and algorithm is necessary to evaluate reference gene candidates. To complicate matters even further, normalization to cell count may not be the most desired option. In this example, we have samples which, for reasons not directly related to a particular treatment of the study, vary in cell volumes. This variation may then overlap any effects observed by the studied treatment. In its normal behavior, we may then observe genes of interest where the expression levels are modulated by cell count alone or modulated by overall cell volume. We may observe reference gene candidates that are proportional to cell count alone or proportional to overall cell volume. Which reference gene to use then comes down to the specifics of the assay and the desired normalization base. This may benefit from some thought in preparation for an assay. Here is a list of reference gene candidates that were evaluated during a workshop that Sigma arranged together with EMBL last year. Samples were collected from a human cell culture in two different treatment groups. I will use this dataset to demonstrate aspects of reference gene validation. I will use two different algorithms to demonstrate the approach in this presentation, and both of them are based on the assumption that a multitude of reference gene candidates can be used to rank the stability of individual reference gene candidates. The assumption may be true if, for example, all reference gene candidates vary stochastically around stable expression levels. However, this may not necessarily be true in reality. To avoid misleading results, it is therefore prudent to avoid regulated reference gene candidates, and in particularly co-regulated reference gene candidates. The list of reference gene candidates shown here was specifically chosen to select genes that belong to different functional classes, which reduces the chance that genes may be co-regulated. A notable exception is GapDH, that is here present in two versions. As we will see in this example, it is of no consequence for our analysis, but best practice is to avoid multiple entries of genes that may be suspected of being co-regulated. The first algorithm I will use is called geNorm. It evaluates gene stabilities by calculating a gene stability measure called M-value based on pairwise comparisons between the analyzed reference gene candidates and all other reference gene candidates in the dataset. It is performed in an iterative fashion, meaning that in our case the procedure is first performed on all 15 reference gene candidates, the worst one is removed, and then the process is repeated on the remaining 14, and the second worst candidate is removed, and so on. In the end, 2 reference genes remain. There may be times when identification of the most stable reference genes may be particularly challenging. One case may be when all reference gene candidates perform poorly. Another case would be if all reference gene candidates perform well. To distinguish between these two cases, it's useful to have a rule of thumb that an M-value below 0.5 may be considered stably expressed. The second algorithm I will use is called NormFinder. This algorithm takes an ANOVA-like approach to reference gene stability evaluation, in that the whole and subgroups are analyzed for variations. One advantage of this is that the obtained measures are directly related to gene expression levels. A standard deviation of 0.2 in CQ units therefore represents about 15% variation in copy number expression levels of the particular reference gene candidate. The bar diagrams shown in this slide were produced using multi-DES data analysis software GeneX. As you can see, both algorithms show reference genes ranked according to their respective stability measures. In addition, I added a graph showing the accumulated standard deviation from NormFinder, indicating that a combination of up to 3 best reference genes may yield stability improvements. As we compare the results of the two algorithms from the current dataset, we notice that they do not agree. This gives us pause to consider the underlying reason. Notice that the worst 2 reference gene candidates in both cases are STHA and CANX. Also notice that the best choice from the NormFinder algorithm is selected GUS-B at this point. Here we see the mean-centered expression profiles of the reference gene candidates of the 2 samples in each treatment group. Samples 1 and 2 belong to the first treatment group, and samples 3 and 4 belong to the second treatment group. As we study the expression profiles, we notice that both STHA and CANX have profiles that deviate significantly from the other reference gene candidates. They affect NormFinder stability estimates of the whole dataset, so that GUS-B, that happens to have an expression profile that to some extent follows the profiles of STHA and CANX, is elevated in the ranking of the most stable reference gene candidates. Due to the deviating expression profiles, we suspect that STHA and CANX are regulated by different treatment alternatives in our assay, and therefore not suitable as reference genes. We remove them from the dataset and repeat our analysis. Now we see that both algorithms agree that the best choice of reference gene is EI4-alpha and ATP53. In the NormFinder calculation of accumulated standard deviations, we also see that the addition of more reference genes beside these 2 do not improve stability. A recent development is the potential use of expressed repetitive repeat elements for qPCR referencing. In primates, which include humans, ALU elements are the most abundant. In mice, B elements are the most abundant. An analysis by next-generation sequencing shows that ALU repeats follows a log-normal distribution, and this is consistent with random incorporation of the repetitive elements throughout the genome. Because of random incorporation throughout the genome, expressed repeat elements are expected to be present in both regulated and not regulated genes. In this example, samples A and B have been collected from 2 different assay groups, such as treated and normal control samples. Gene 1 in red is unaffected by assay conditions. Gene 2 in blue is upregulated in samples B compared to samples A, and gene 3 in green is downregulated in samples B compared to samples A. Even though expression of B1 elements is different for each gene, the overall expression of B1 elements is stable, in this example at 6 expressed B1 elements. Since expression of repeat elements samples expression of many independent genes in this way, the overall expression of repeat elements may be a stable indicator of overall level of transcriptome, and may thus be the closest alternative we have of a universal reference for qPCR. However, absence of bias cannot be taken for granted, and a validation against a panel of reference gene candidates is strongly recommended. In this graph, reference gene candidates were evaluated by geNorm for an assay of human samples. 2 commercially available reference gene panels were used. In addition, 2 versions of allorepeats were tested, allo-SX and allo-J. With the majority of candidate genes tested in duplicates, it is believed effects of validation bias was minimized. As can be seen in this example, the allorepeats show great promise as stable references in general, and they are the most stable references for this assay. To summarize, use a panel of reference gene candidates to validate your reference gene. A reference gene on its own is not reliable. The reference gene candidates need to be independently expressed. Using both geNorm and NormFinder allowed us to cross-validate the validation methods. We saw that expressed repetitive repeat sequences may be interesting new targets as references. Consult Sigma experimental design and data analysis at www.sigma.com/designmyprobe for additional support and answers to direct questions. We enjoy being a small part of your scientific project, and we offer free personal advice on many qPCR topics. We have online tools available directly at our site, and we can also perform personalized services for you. We are happy to assist you regardless of what product you are using at the moment. While planning future experiments, have you considered Sigma's range of qPCR reagents and kits? Use a combination of Sigma services and products for guaranteed success. This was a brief overview of qPCR reference gene validation. I hope you enjoyed it. Look out for more presentations in the Sigma-Aldrich series. Thank you for your attention.